Vegetative propagation can occur in numerous ways, which are outlined in the germplasm guidelines. One of the most common is semi-hardwood cuttings of current or last season's growth. In this video, Mandy Thompson from the Royal Botanic Gardens Victoria, Cranbourne, explains the method.
So we enact the hygiene protocols. So we do a physical inspection of the pest for pest and disease. We'll drench in detergent and also in eucalyptus disinfectant with water and rinse clean before we start working with the material. And so clean methylated spirits and sharp secateurs and a sterile environment wherever possible. Sometimes in the field that's not possible, but in the nursery it is. We just process that cutting material down into a cutting that will fit into this preformer plug, which creates 100% capillary action from the moment the cutting goes in. We generally wound the cutting just below a node where it's going to grow, and then we'll process that cutting material down just to reduce the transpiration that we might get. So we'll use just a hormone like this Clonex purple, and that's indole butyric acid, and it's just a bit of a dip in there, and then place that into the plug. We use the plugs because it reduces transplant shock, as well as keeping 100% capillary action and keeping them moist. So you can imagine when that's struck and it goes into the tube, into a completely different media, a completely different environment, that you can reduce one of those factors by keeping it snug in its plug still, so you're not disturbing the roots too much. So that's the reason why we use these. They're not for everything, they're not for a lot of desert flora, but most of the material we grow here at the gardens can get through really well in a plug.
Once the cuttings have been processed, they are maintained in the nursery glasshouse until roots develop. Once roots have developed, cuttings can be transferred into forestry tubes with pasteurised potting mix appropriate to the species and the final planting location. For more information on this process as well as other propagation techniques, please see Chapter 8 of the Germplasm Guidelines.